biomedical technique in cancer research and diagnostics and disease monitoring. That is flow cytometry. So this presentation I have divided it in 4 parts. The first part will be about the introduction. Second part will cover the basics of data analysis, which will here I will just explain about the data plots and getting. And third plot will cover the activated cell sorting, that is most important for most of the students. And fourth part is applications. So let's have a look at the introduction parts. So the flow cytometry is a biomedical technique which is used to analyze and sort the population of cells from a heterogeneous population of cells based on their light scattering properties. So if you have 2-3 populations of cells in a single tube, where you can analyze those cells using flow cytometry. So for example, in this plot you can analyze CD4 and CD19 cells at the same time. So what is flow cytometry? The flow cytometry you can divide it in 3 parts. The flow means sheet fluid or cells in motion. Cyto is cells and metry is measurement. So the flow cytometry is a technique where you analyze the physical and chemical properties of cells based on their light scattering properties. So the technique is extensively used in immunological studies and diagnostic purposes. And this technique is quantitative method because here you can analyze up to 1000 cells per second, and it's a multi-parameter instrument. I will just quickly brief you about the history of flow cytometry. In 1953, Wallace H. Coulter, who developed first impedance-based flow cytometry device using Coulter principle, and Mark Fallweiler is the inventor of today's cell sorters. And in 1968, Wolfgang developed a first fluorescence-based flow cytometry, which is ICP-11, and the name FACS is owned by BD. The FACS means fluorescence activated cell sorting, and before it was termed as pulse cytophotometry because the pulse is voltage pulse, cells, light and measurement. So later they termed it as flow cytometry. You will come to know why they termed it as flow cytometry. Let's have a look at the principle of the flow cytometry. So when a suspension of cells are passed through a flow core, this is the flow core, where with the help of hydrodynamic focusing, the cells will pass through a small orifice, a narrow orifice, one at a time. These cells will hit the laser beam here, and which scatters the light. This will scatter the light. The scattered light is collected by the detectors placed in front of them. So these are the detectors which are placed in front of the laser beam, and the detectors will convert the photon data into electronic signal. That electronic signal is converted into digital data, which is recorded and analyzed by a specialized flow cytometry software in a specialized computer. Okay, so next, components of flow cytometry. So there are 4 main components for a typical flow cytometry. So first one is fluidics part, which involves the sheet fluid, flow core and hydrodynamic focusing of the cells, and second, which involves excitation lasers and emission filters, excitation lasers and filters, and detectors, which involves the photomultiplier tubes which collect the scattered light and they convert the light signal to electronic signal, and this electronic signal is converted in the form of digital data. And the computer system, which is connected to the flow cytometer, has a specialized flow cytometry software which is required to record the data and analyze. So first let's have a look at the fluidics part. So the fluidics part of a flow cytometry involves hydrodynamic focusing. That is why I have highlighted this one, hydrodynamic focusing, because this is the most important thing you need in flow cytometry. So the hydrodynamic focusing of the injected suspension of cells in a flow cytometer to properly analyze cells or count the number of cells, then they need to pass through a narrow orifice one at a time here. So hydrodynamic focusing uh, is achieved by the sheet fluid. This is cell suspension, this is sheet fluid. This will help the cells to pass through a narrow orifice one at a time. So what happens is hydrodynamic focusing solves the problem by building up the walls. These are the walls, sheet fluid walls from fluid using the effects of fluid dynamics. Okay, so the white tube made up of glass or plastic, this one is used, through which a wall of fluid is called sheet fluid is pumped. This is the sheet fluid, so which is pumped. So here the cells can align one at a time. The sample is injected in the middle of the sheet fluid. This is the sheet fluid where the sample is injected in the middle here. So the velocities and velocity difference is achieved so that these two this cell suspension and sheet fluid don't mix up and clog the instrument. So here they will the hydrodynamic focusing will allow the cells to pass through an orifice one at a time. So then they form a two layers stable flow. That's why the sheet pressure is different from sample pressure. So usually the sample pressure is 3 psi over sheet pressure. Here you can see the cells are aligned in such a way that it will pass through a narrow orifice one at a time, then they hit the laser beam. So optics part. So the optics part of flow cytometry involves the excitation lasers. For example, you might have heard of blue laser, red laser, violet laser, and emission filters such as band pass filter, short pass filter, and long pass filter. So and there are also dichroic mirrors. I will explain all of them. So the lasers are used as excitation sources which excite the fluorochrome attached with the cells, and they are used to measure the autofluorescence of the cells and fluorescence of the cells. So filters are used as emission sources which will pass or block the light wavelength through them in the specified or cutoff range. So emission filters which are involved in flow cytometry are band pass filter, short pass filter, long pass filter. So the short pass filter, this one, the short pass filter will allow the light wavelength below cutoff range. For example, here the short pass filter is 500, that is SP 500. So which which will allow the light wavelength below 500 nanometer. So this one will allow the light to pass through the filter below 500 nanometer, and above 500 nanometer it will block it. This is a long pass filter, that is LP 500. Here what the long filters will allow the light wavelength above the cutoff range. For example, this is LP 500. Here 500 LP is a long pass filter which will allow the light wavelength above 500 nanometer. So below 500 nanometer it will block. And this is a band pass filter. The band pass filters allow the light of specific wavelength in a specified range. They have the specific range. For example, here is 500 by 50 is a band pass filter. So this 50 nanometer is divided in minus plus, that is minus 25 plus 25. So what is it actually? So the is a which will allow the light. This will allow the light within the range of 475 and 525 nanometer. Don't get confused about the band pass filter. So the 500 divided by 50 means it will allow the light within the range of 475 to 525 nanometer, and it will block the rest of the light. There are also dichroic mirrors which are used to reflect some of the light and transmit the rest of the light. So these mirrors are also important in flow cytometry. So the dichroic mirrors are the one which will reflect the light in 90 degree they are actually placed in 45-degree angle, so that the incoming light will light is reflected in 90-degree, and the rest of them is transmitted. For example, this is a 540 nanometer dichroic short pass mirror. So here what happens is, the dichroic mirror is placed in 45-degree angle, so the light, and it will the light source, the dichroic mirror will reflect the light below 5 will reflect the light above 540 nanometer light, above 540 nanometer. I'm sorry, I will explain it again. This is a dichroic mirror. So this is a short pass 540 nanometer dichroic mirror. So the short pass, you know what about the short pass, but the mirror is different. So here the dichroic mirror is placed in 45-degree angle, where the incoming light will reflect the light above 540 nanometer, and it will transmit the light below 540 nanometer in this example. Got it? Okay. We'll go to next. Okay. And the dichroic mirrors are installed before emission filters. Here in this schematic image I will explain you. This is the here the cells will hit the laser beam, will scatter. These are the specific filters, and this is the dichroic mirror. Okay, so here the 90 the some of the light is reflected and some of the light is transmitted. Okay, this is the emission filter. So the emission, so what happens is, when the light source hit the dichroic mirror, so it will reflect the light in 90-degree. The 90-degree reflected light is collected by a specific filter which is placed in front of the PMT detector. Okay. So detectors. So the forward and side scatter light and fluorescence from stained cells are split into defined wavelengths and channeled by a set of filters and mirrors within the flow cytometer. So the fluorescence light is filtered so that each sensor will detect fluorescence only at a specified wavelength. These sensors are called photomultiplier source. So that is the definition I told you about. Okay. So here I will explain in this schematic diagram. So when the cells hit the laser beam, these cells will scatter the light in all directions. So the light scattered in forward direction is collected by a detector called forward scatter detector, and this detector will give you the approximate size of the cell, and the light which is scattered in to the side, where the side scatter detectors are placed in 90-degree. So 90-degree. So here 90-degree. So the side scatter will detect the granularity of the cells. Okay, and along with the side scatter there are fluorescence detectors. Okay, so I will explain it again. Okay, so when laser beam hits the cell, the cells will scatter light in all directions. So light scatter in forward direction is called forward scatter, which is measured by detectors placed in front of the laser beam, and measures the approximate size of the cell. And the light scattered to the side about 90-degree is called side scatter, which is measured by the detector called side scatter detector, and it measures the approximate granularity of the cell. Along with the side scatter detectors, there are fluorescence detectors which measure the fluorescence emitted by the cells. Okay, so here is an example. So side scatter, this is SSC, side scatter, forward scatter. These are the 4 different populations. Here these are the debris. So this is the blood cells, where lymphocytes, monocytes and granulocytes. These are giving populations based on their size and granularity. Okay, then we will move to the next. Measurement of signal. So as the fluorescing cell passes through a laser beam, it creates a peak of photon emission over time. So these are detected by the PMT and converted to a voltage pulse known as event. Okay, the total pulse height and area is measured by the flow cytometer. So the measured voltage pulse area will correlate directly to the intensity of the fluorescence of that event. In this figure you can see the PMT measures the pulse area, the voltage created, and each time a fluorescent, so here this one. Okay, you can see when the cells start passing through a laser beam, it creates a peak emission like this. The moment it leaves the laser beam, it yes, that's what I'm telling you. Okay, got it. Okay, and one more thing, if there are no cells, no event, no nothing. Okay, so before I go to the data analysis part, I will just again I will give you the brief introduction. Briefly I will just describe this schematic diagram. So when you give the cell suspensions, it will go to the flow core. Here the flow core, because of the hydrodynamic focusing of the cells will pass through the nozzle one cell at a time. These cells will hit the laser beam, and the laser beam, that's what let me explain first. So okay, the cells will scatter the light in all directions. The light scattered in forward direction is called forward scatter. This is collected by the forward scatter detector, and the light scattered to the side is detected by the side scatter. Okay, and these are the dichroic mirrors, emission filters, and these are the fluorescence channels. Okay, the detectors will convert the photon energy into electronic signal. That electronic signal is converted to digital format, where using computer or CCD you will record the data. Okay, so let's move on to data analysis part. So in flow cytometry you can analyze multiple fluorochromes in a single run. For example, you might have heard about the multicolor experiment, which involves the analysis of 4 to 10 fluorescent signals from the same samples. Okay, the output of the detectors can be displayed in number of different ways. So the flow cytometric data is in the form of events which are generated by the voltage pulses. Just before I explained you, voltage pulses, and collected by the PMTs, which convert the photon signal into electronic signal. So each dot or event represents one event in flow cytometry plot. Using flow cytometry data, you can quantify the number of cells. You can see how many cells are there, and respect to fluorescence emitted, and the data allows you to select the population of interest using gating tools, and using these you will get the mean and median fluorescence of the intensity, which you might have heard of. So there are 4 different types of plots in flow cytometry: dot plot, density plot, contour plot, and histogram plot. So the histogram plots are used to check the shift in fluorescence signal from here to here, how much fluorescence signal is shifting from one sample to another. And this data analysis part I will cover it in the next presentation, because I will give the live demo of the data analysis. Okay, this one I am just giving you the overview. The next presentation I will give you the complete data analysis part, live demo. Okay, so the gating population of interest, there are various types of gating tools. Some of them are rectangular gate, and polygonal gate, eclipse gate, quadrant gate, and interval gate. And these 3 are for the population, to select the population of interest. Here this is a dual parameter where you can see the 4 different population of cells, and this one is interval gating to get your fluorescence signal of interest. Okay, so fluorescence activated cell sorting. The FACS is a specialized type of flow cytometry which analyzes the cell and can sort the population of interest which are collected in a collection chamber or tubes. Okay, so this technique provides the sorting of heterogeneous population of cells into two or more containers based on their light scattering properties and the droplet charge. It provides the quantitative recording of fluorescence signal from cells as well as physical separation of cells based on their fluorescence signal. So basically the population of interest from a sample tube are sorted, and these sorted cells are used for downstream experiments, for example RNA isolation or cell culturing, and some students do microarray analysis. So let me explain you this one principle. So this is the schematic diagram of the cell sorter. Okay, so what happens is when you give the cell suspension, there is an ultrasonic nozzle along with the vibrator. Okay, ultrasonic nozzle vibrator, okay, which forms the droplet of cells like this. Here you can see the droplets. So these droplets are charged. Okay, these droplets are charged depending on, it depends on you. Okay, so the cells are passed through a nozzle assembly where cells are hydrodynamically focused one cell at a time. So this will pass through the vibration mechanism is installed near a nozzle, this one. Okay, where the stream breaks into droplets. Yes, at a specific frequency, and this system is adjusted so that there is a low probability of more than one cell per droplet. Okay, before stream breaks into droplets, the cells will pass through a fluorescence measurement, so where the fluorescence is measured, and an electronic charging ring is placed just at the point where the stream breaks into droplet. The charge is placed immediately prior to the fluorescence measure, and the opposite charge is trapped on the droplet as it breaks. The charged droplets are the droplets containing cells then pass through the electronic deflection plates which divert the droplets into collection tubes. Okay, sure. I will explain it again. Here in this image I will explain again. So here you are giving the population of cells, cell suspension. So the sheet fluid will this you know, right? Yes, with the help of hydrodynamic focusing, the cells will pass through the nozzle orifice, nozzle both you can see. So small nozzle, one cell at a time. So there are some vibration mechanism and this one electronic charging ring, electric charging ring. So this electric charging ring will place the charge on the droplets before prior to the fluorescence measurement. Okay, so and let's suppose you have 4 different populations to sort at one single time. It will give you the charges based on like minus, negative, positive, like left, far left, right, far right, like that. You can, using the deflection plates, it will sort the cells in different containers, and the waste will go to the waste which you don't need. It will directly go to the waste. Okay, and this is an example of 6-way sorting. This is in influx sorter. Application part: immunophenotyping. This is an example data of immunophenotyping where you can analyze or sort different CD markers. Apoptosis, where you can study the apoptosis and necrotic cells, In this one you can say early apoptotic or late apoptotic. Cell cycle analysis, you can analyze cell cycle, and calcium flux, gene expression, green fluorescent protein or yellow fluorescence protein. And other applications are cytokine analysis, cell counting, DNA damage, kinetics. The flow cytometry is also being used in aquatic research, microbial species identification. So in next presentation I will give the live demo of data analysis, and after that presentation I will give the live demo of cell sorting. Yes, in cell sorting I will completely give how it works, and sample preparation guidelines, and what are the controls used in flow cytometry, what you should do before you plan your cell sorting experiments, and how you can analyze cell cycle and multicolor flow cytometry.